Thank you. Hello and good morning, everyone. I'm Farnaz Delavari. I'm a medical doctor, and I'm currently doing my second year of PhD in the lab of Professor Elias in Geneva, Switzerland. And today I'm going to talk about my ongoing research about the neuronal substrates in the 22q deletion syndrome, which in this case today is the abnormal amygdala functional connectivity.
Recording in progress.
So as you probably all know, and it has been discussed also yesterday, the 22q microdeletion is a microdeletion in the long arm of chromosome 22. And although it's a microdeletion when compared to the non-deletion carriers, we observe a huge diversity in the clinical profile, and more specifically to this talk in the brain abnormalities and psychiatric disorders. So you can see up to 40%, 30% of the population with 22q experience ADHD, anxiety disorders, and depressive disorders. But a bit more specifically to this population, because the other disorders are also observed more as comorbidities in other neurogenic disorders. What is more specific to this population is this schizophrenia-like psychotic disorders, which occurs up to 30 to 40% of the population, and it's much higher than the general population. So this is the main interest of my research, actually, to see, okay, so 30 to 40% of the population are developing psychotic disorders, but why is it that despite of all patients carrying this genetic deletion, some don't develop serious psychopathologies, thankfully. So this all brings the question of resilience, as was discussed in the previous talks. And of course, this deletion has a developmental impact. So an important factor, in addition to the genetic disposition, would be the environmental factors. And both of these genetic and environmental factors are currently being discussed that are easily, not easily, but what I mean is that one of the common means to assess these factors is the neuronal substrates. So naturally, we are interested in the neuronal substrates of susceptibility to psychosis and the predictive markers. Now, findings in behavioral analysis, especially in our lab, has been shown that anxiety and affective disorders such as depression are common disorders in children with 22q and have a great impact on their adaptive functioning. And then they have gone further to show that the presence of anxiety disorders in children and adolescents with the 22q is a major risk factor for the emergence of psychotic symptoms. And in a very more recent study by Dr. Sandini, who presented yesterday, it has been shown that actually impaired tolerance to stress is in the follow-up of the patient is a funnel symptom that is very greatly affected by the baseline symptoms of these patients. So we observed this the time trend of anxiety and psychosis in the behavioral analysis that could be both a risk factor that can be interpreted as a causation or just a prodrome or both. And we are given this prior research that point to affective and anxiety symptoms playing a role in prediction of developing psychotic symptoms. We are aiming into looking into this neurobiological structure of associations that are most associated with emotional regulation. And as you can see, amygdala is known as one of the key structures in affective and processes involving anxiety, pain, and emotions. And also it has very distinct functional sections. But despite this, amygdala has been very well studied in anxiety disorders, but not so much in psychosis, especially for the subsegments of it. And this is something that we were really interested to see how the subsegments and correlations with other brain regions change across time. So to do this, we use the functional MRI, and it's a resting state scan. So we ask the participants to lie and relax in the MRI machine and look at the black screen with a white cross on it, and just let their mind wander, not to fall asleep. So it's an easy task for children also to do, and we have samples from 5 to 35. And then we look at the oxygenation level of the proxy measure of oxygenation level of the brain regions, and we see which regions activate and deactivate together. And we say, okay, so probably these regions are connected. We have, thanks to the amazing families, we have 191 participants, up to 300, almost 300 longitudinal scans. And we were able to divide the participants based on having mild to moderate positive psychotic symptoms into patients who are at a higher risk for developing psychosis and lower risk. And then we used maximum likelihood measures to find regions that are functionally distinct within the amygdala. So with this method, we found 3 regions: basolateral amygdala, centromedial amygdala, and superficial amygdala. So I will walk through the results and the interpretations that we have so far very fast. We have observed that the BLA, or basolateral amygdala, which is a seat that is in the amygdala known as the inlet scene, and it's known to has to have strong connections to cortex to perform well. Here we see that in patients with 22q, there is a lack of associations with regions in blue that are actually prefrontal regions in charge of emotional regulation. And at the same time, we see an hyperconnectivity of this seed region of basolateral amygdala with hippocampus and parahippocampal regions, and which are basically regions that are in charge of memory and internal processing. And interestingly, this pattern is a pattern that has been observed in the literature for patients with PTSD and phobia. And what we observe is that there is no age relationship with this pattern, or at least in the age span that we have, which is 5 years old, we cannot observe it. So it happens early on, this deficit, and is almost stable with age. One thing that is noted importantly is that, okay, so we see that there is this PTSD pattern that is also associated with PTSD in other studies and phobia. And we know that a specific phobia begins very early in our patients and is represented through all age groups of our of patients with 22q. Of note, I would mention that in another separate project, we actually are confirming that actual white matter fibers between hippocampus and amygdala and prefrontal are changed with the 22q deletion. So this confirms our functional results. As for the other, next I would point to the results for the superficial amygdala, which is mostly consistent of glutamatergic neurons. And in this region, we see that there is an abnormal strong connection with the striatal regions and prefrontal regions. And this is specifically more strong and gets stronger with aging in patients who later develop positive psychotic symptoms. And because it's this region that is mostly consistent with the glutamatergic regions, we hypothesize that probably superficial amygdala participates in this hyperdopaminergic state that is observed in patients with positive psychotic symptoms. And I would note that this age relationship happens very early, like normally earlier than we see the age. So it's here around 10. And we would point that this could be an early biomarker that we can investigate more. Then the last section is the centromedial amygdala, which is mostly an outlet region for the subcortical brain. So it's where amygdala points to the subcortical regions. And then here is when we have projections to hypothalamus, to adrenal axis, and we have this fight and flight response. And we see that abnormally in patients with 22q, there is a weakening of connections with higher order cortices that have this regulatory role and some of the sensory regions. And we see that this weakening is especially happening in patients who develop psychotic symptoms, positive psychotic symptoms. And we hypothesize that this probably is in charge of transforming the lack of upper control to the hypothalamus and adrenal axis. And which is interesting because in our patients, it has been shown that hypothalamus adrenal axis is impaired. And this is also corresponded with impaired tolerance to stress. And interestingly, in amygdala, this was the only pattern that also we find a correlation in patients that had impaired tolerance to stress. So both patients who had impaired tolerance to stress and psychotic symptoms developed this weakening of centromedial amygdala with prefrontal regions. And it is worth to note that this centromedial region is highly affected by cortisol. And it has been many studies showed in resilience studies to stress that having an optimal connection between centromedial amygdala and prefrontal regions is dependent on corticosteroid receptors and is necessary for an optimal response to stress. So if I want to summarize my key findings, I would have to say that we have shown that distinct regions within amygdala with different age patterns and different behavioral implications exist in the 22q, and these could be different neuronal biomarkers. One be diagnostic specific, like the basolateral amygdala, which did not find the behavior or correlate for psychosis, and the other two more specific to psychotic symptoms. So this is very interesting for us because we see this combination of age-stable or early biomarkers and later changes in the brain. It would be interesting to later see the internuclei connectivity and how these nuclei affect each other. If one nuclei impairment happens first and then causes the next one. And also because we have results that point toward the impairment of cortisol secretion, we are very now interested to see if we can find any correlation with these amygdala abnormalities and the impairment of cortisol response. Thank you so much for your attention. I think I was 1 minute off.